Good morning. So we are starting a new section now where we are going to start talking about chromosomes. Because as we know, our chromosomes have genes on them, and now we need to learn how to pass on those genes. Um, so we're going to start in our first section talking about generalities of passing on your DNA. So, our genes are basically individual segments of our DNA, our deoxyribonucleic acid. And a gene is a specific area that's going to code for a protein through that process of where we have transcription and translation. And we're going to make a protein that has a specific function. And that's the whole point of a gene. And so, if we go back to our DNA, most of the time it's just free floating strands in the nucleus of a cell. It's not wound up tight. They're just long strands, about 5 feet long, thinner than the thinnest hair that you know. And it's known as chromatin. And this is how it sits in the cell the vast majority of the time when the cell is in a specific phase called interphase, which I'll be talking about in another video. During this time, the DNA is available to be used through the processes of transcription and translation. So as well as for DNA replication if needed. And so with transcription and translation, we can start making amino acid chains. So we can make proteins and macromolecules that the cell might need. So if we want to know what a chromosome is, chromosomes are tightly packed DNA. So we start with that DNA double helix just floating around as chromatin. And what's going to happen is it's going to wrap itself around these proteins that are in the nucleus of the cell called histones. And so the DNA wraps around the histones and then it continues twisting around it. And just like if you would take a rope and you would twist it, twist it, twist it, it would shorten and fatten. Well, that's what's going to happen. It's going to shorten and condense and condense and condense. And eventually it, excuse me, becomes a visible chromosome. And these are only found during the process of cell division. So during the vast majority of the time when a cell is doing its work, we just see chromatin. And the reason for that is when the DNA is all twisted and condensed up, it can't be used for cellular processes. Now, we have a terminology problem in science in that we use the same word chromosome for what could be considered a singlet as well as what could be considered a twin. Because when people look at this X-shaped thing, we call that a chromosome. It's really, think of it like a conjoined twin. Um, so this single thing right here, that's a chromosome. Okay. And when that chromosome gets duplicated, that gives you the X-shaped thing. So a duplicated chromosome, we also call a chromosome. And in a duplicated chromosome, we call each of the singlet things a sister chromatid, except when we only have one sister chromatid by itself, we can't call it a sister chromatid because there's no sibling around, so we call it a chromosome. So yeah, the terminology can be a little bit confusing. So a sister chromatid is one of the two attached members of a duplicated chromosome. So I'll try to be really clear when I'm talking about chromosomes, and you'll know if I'm talking about like the little singlet chromosome. But otherwise, most of the time when we're going to be talking chromosomes today, we're going to be talking about duplicated chromosomes. What you notice in the middle of each chromosome, there's this little area called a centromere. And it's in this centromere, this is the place where the sister chromatids are going to be attached to each other. So yes, the centromere does exist in the single chromatid by itself as well as in the duplicated chromosome. So, as you probably know by now, humans have 46 chromosomes, or what we say 23 pairs of chromosomes. Out of those 23 pairs, and here's an example of a karyotype of somebody in a cell, somebody's 46 chromosomes. Here in the pale lilac background, you can see 22 pairs of what we call autosomes. These are the chromosomes that are not involved in determining the biological sex of the individuals. And you can see that there's 2 chromosomes for each pair. And so basically you get one from the mom and one from the dad. So we say there's a maternal chromosome and a paternal chromosome. And they are considered identical. We're going to use the word homologous, but I'll introduce that term on a later slide. Then we have another pair, but the pair may not match each other. These are the sex chromosomes. If you are a female, then they are a match set because you would have 2 X chromosomes. But if you are biologically a male, then they would not match each other. They're still a pair because you have 2 of them, but you would have an X and a Y. So the number of chromosomes basically has nothing to do with the size of the organism or how complex the organism is at all. Um, Humans have 46. As you can see, a dog has more than we do. Cats have less than we do. And if we look over time with evolution, the number of chromosomes can change. If we look at how the evolution of the wheat species changed over time, and we look way back 90,000 years ago, we can see that there were 2 different species, each of which had 14 chromosomes, and when they were able to breed with each other, they made a wild type that had 28 chromosomes. And that was able to breed with another goat grass that had 14 chromosomes. And instead of adding 28 with 14, they were able to make a species that had they were able to make a species that had 42 chromosomes, but they were also able to make a species that only had 28 chromosomes. And so eventually between over 30,000 years to about 8,000 years ago, we have our modern wheat, and we actually have 2 different types of wheat now. We don't really use any 14 chromosome wheat. Those are all considered grasses now, but we have durum wheat, which has 28 chromosomes, and a different type of wheat has 42 chromosomes. And if you are a wheat aficionado, you know that durum wheat typically is the type that is used for semolina, for pasta flour, whereas the bread wheat, a totally different type of wheat, is used regular flour. So very interesting stuff. It happens over time. So that brings me to another concept we need to talk about, which is ploidy, which has to do with the number of complete sets of chromosomes in a cell. So as I told you, humans have 46 chromosomes, or 23 sets, okay? So we say one set of chromosome is N, and so 2 sets of chromosomes would be 2N. So if we have 46, which would be having 2 each of 23 different chromosomes, that would mean we would have 2N chromosomes if we have 46 chromosomes. That is the same thing as saying we were diploid. Okay, so here's an example with an organism that wouldn't have 46 chromosomes, but would only have 3 chromosomes. So if it has 3 little single chromosomes, we would say it's haploid. but then if it has a maternal and a paternal one of each of them, we would say it is 2N or diploid of it. And so here it has 6 chromosomes or 2 each of 3 different sets. So our non-gamete human cells, our autosomes contain our non-gamete human cells contain 23 homologous chromosome cells. They are all diploid. They have the 46 chromosomes, where one comes from the father and one comes from the mother. So that word homologous, which I threw in here for the first time, means similar features. So what does that mean? Well, what it means is whatever gene is on this chromosome over here is on the other chromosome over there. So in other words, the moms are giving the gene at a certain place on this chromosome, and the dads are giving the genes on the same place on the homologous chromosome. Now, our gamete cells, in other words, the sperm from the males and what you may like to call the egg from the females. Okay, but we're not chickens. We don't lay eggs. We have oocytes from the females. These are our gametes. They are haploid. And the reason why they are haploid is so that the dad can contribute one and the mom can contribute one. And so when they join together, then the offspring will be diploid. Because otherwise, if the mom contributed 2 and the dad contributed, the offspring would have 4. And that would be a problem. Okay. So by our gamete human cells being haploid, we are ready for reproduction to occur. Okay. So our haploid or N cells are considered non-homologous because they're not pairs. Where our diploid 2Ns, where we have a copy of each chromosome, these pairs are considered homologous chromosomes. All right. So in our human life cycle, we started from a single cell, and we were most of our cells were 2N, and they grew and divided into adults ready to reproduce, at which time the gamete from the male, the sperm, and the gamete from the female, the oocyte, which were haploid, joined together, and the sperm fertilized the oocyte and ended up with a diploid offspring. And that's how the human life cycle continues. All right, so I'd like you to end with this picture. I've indicated some things for you to practice labeling, and I'd like you to use all these terms and see if you can explain all of them. And that's it for this section, and I will see you shortly when we continue and I talk a little bit about the life cycle. Thank you so much for all your work in this course.